Today we discuss about our chapter molecular basis of inheritance. In this chapter we discuss about the genetic material is DNA. DNA. In early 90s, the scientists which are related with the genetics, they told that the DNA is a genetic factor. The DNA is present inside the nucleus. In previous chapter we discussed about this cell. Inside the cell we found a nucleus. This is the nucleus. And inside the nucleus this chromatin material is present. Chromatin network is present, and inside the chromatin, that is also we call it as a chromosome. And the chemical composition of chromosome is a DNA and protein. Right? This is the early in early 90s. The scientists told that the DNA that is the nucleus. Inside the nucleus we found the chromatin network. Inside the chromatin network we call it as a chromosome, and that chromosome is chemical composition of DNA and protein. And this DNA is carrying the genetic information. DNA is carrying a genetic information. Which type of information? That are the skin color, eye color, hair color, which are carrying from his father to the son or grandfather to the grandson. Okay. Then on this DNA there is a presence of genes. There is the presence of gene. And these genes, what is the function of these genes? These genes carry characters. On this gene, what does DNA do? DNA carrying certain characters like hair color, eye color, skin color. All those characters are present inside this chromosome, right? This is called a DNA in the early 90s. But before that, scientists told that this protein is a major factor of which carrying before 90s, scientists told that, or geneticists told that the protein is a major source of what? Major source of transporting characters, major source of doing all metabolic activities carrying inside the cell, and that the protein is responsible for that. But after that, the scientists done various kind of research on this, and they finally conclude that the DNA is a major factor present inside the nucleus. And this DNA carrying various kinds of function, DNA carrying various kinds of function like carrying genetic information, like doing the helpful in protein synthesis also. All those things are doing or carrying with the help of this DNA, and DNA is a large quantity inside the particular cell, which is carrying character from generation to generation. That is the main source of this point, that is the genetic material is a DNA. Now our next point is Griffith's experiment. Griffith's Griffith experiment. In 1928, a medical officer, that name was Frederick Griffith, he done an experiment on bacteria. Which bacteria then? That is a Streptococcus pneumoniae. That is a Streptococcus pneumoniae. What is that the role of Streptococcus pneumoniae? That is, this Streptococcus pneumoniae causes a pneumonia. Because of that, the number of patients are die due to the causing of that disease, name is pneumonia. Okay. For that, he used two strains or two genetic varieties of Streptococcus to find a cure of pneumonia. He want to do a work. We want to research. You want to research on this disease causing element. And what is that? That is the Streptococcus pneumoniae. For that, he take two strains. What are those? Look here. That is S strain or smooth walled. S strain or S, we can call it as a S type. What do you call it? S type. That is smooth walled, right? And they are virulent. They are virulent in nature. Virulent nature means they are disease. They are having a disease causing ability, or they causes a disease to the particular human being, right? And these are the smooth walled. Why they are smooth walled? Because they having a outer layer of polysaccharide. The outer layer of polysaccharide layer. Due to that, this S-type bacteria, they are smooth wall. How they look like? If this is S-type bacterium and this is the outer layer of polysaccharide. That's why this is a smooth walled. Then he take R type of bacterium. R type bacteria. We take R type bacteria. They are the non-virulent in nature. Okay. Non-virulent in nature because they are not doing any give any harm to a particular person. That's why they are non-virulent in nature. Why they are rough walled? Because the outer layer of polysaccharide is absent. Outer layer of polysaccharide is absent. That's why they are rough. They are like this. There is absence of this polysaccharide layer or mucus layer, which is or coating. We call it as a coating also. What is the function of this coat? Why this coat is essential for this S type? Why we call it a smooth wall? All these type of questions are solved in our Gibbs experiment. How this polysaccharide is very essential to this S type bacterium. Okay. Then he take what he do? He take a S type bacteria. He take one second. S type bacteria or S type, we can call it as a S type. S type. He inject into a mice. Inject into a body of mice. What is the result of that? The result of that, mice is die. Mice become die because this is S type bacteria is a virulent bacteria. Because of that, these bacteria having a pneumonia causing factors. That's why this when Griffith inject in the body of mice, the mice is died because of the pneumonia. Okay, he suffered from pneumonia and died. Okay, then he done another type. He take another type, that is R type. Inject into mice, and that mice is live. Mice is live. Mice is not dead because this R type bacteria is non-virulent. This is not causing any type of disease or pneumonia. This R bacteria, it doesn't have a pneumonia causing element. That's why this mice is alive. Okay. Then he take a S type bacteria. S type, and what he done? He hit and kill. What he done? What he do? He hit and kill. And what is the process of this hit and kill? Why he want to hit and kill the virulent nature of this S-type bacteria. That's why he give a hit and kill process, and that hit kill S-type bacterium he again inject in another mice. What happened here? Here also the mice is alive. Mice is alive. Why? Because this S-type bacterium having this virulent polysaccharide coating, their polysaccharide coating is removed. Why is removed? With the help of this heating killing process. Due to this heating killing process, this virulent nature of the S type of bacteria is removed, and because of that this mice is alive. Okay, understand? Okay. Then he done another experiment. What he do? He take S type plus heat kill like this. He repeat this process, and again he take R type bacterium. Look here, what he do? What he take? He take this S type bacteria which are already we take here, heat kill, we remove the polysaccharide coating, and due to that this mice is alive. Then he think, when I added the Griffith think that when I added this into the R-type bacteria, we mix with the R-type bacteria, what happened? He get a very shocking, he inject into mice. What happened here? Here, a very shocking experiment, he found that mice is died. And literally, he shocked. Why this mice is died? For that, because he observed the virulent nature of this S-type bacteria is completely removed, and we can observe here also. Then this R-type of bacterium, it doesn't having virulent nature. Then why this mice is dying? Right? Then one question is arises in our mind, that is, what about the immune system? What about the immune system of this mice? What about the immune system of mice? Because of that, he doesn't alive, or he is dying. At the time, the antigen and antibody interaction is not done there. Okay? What happened? Antigen and antibody interaction is not done. That's why mice is died. Understand? Okay. The immune system is every human being having his immune system. And like that, the antigen and antibody interaction is done, and on the basis of that, the human beings or mammals, they are alive, or they are survive with the help of, they are survive. Okay? Then Griffith conclude that this S-type bacteria having hit kill, this coating is this S-type bacteria is come over to this R-type bacteria, and he done this type of behavior, or this polysaccharide chain is formed there. Griffiths conclude that is there any type of substance or any type of nature is or genetic property they are transporting, they are after hitting killing process also these properties alive there. Because of that, this coating is overcome towards the this R type of bacterium, and because of that this mice is die. Okay, this is about the Griffith experiment, but doesn't explain give an explanation about the which factor is required, or which because of which factor this mice is die. He is unable to give explanation about the Griffith experiment. For that, we need another type of we having inside our book, we having another type of experiments, and all that experiment we will be talk about in our next lecture. Thank you.